Bismillahirrahmanirrahim. Assalamu alaikum. Today my topic is hybridoma technique. Hybridoma technique is an emerging technique which is used very frequently nowadays. After discussion at this topic, we will learn about what is hybridoma technique. We will also see the history, how hybridoma technique works, how monoclonal antibodies are produced, its procedure, what is HAT medium, applications of this technique, and at the end, inshallah, we will discuss about the brief summary of the hybridoma technique. First of all, what is hybridoma technique? As its name shows that in this technique, we use a hybrid. Hybrid is a thing that are produced after the mixture of two things. Its definition is that it is a method for producing large numbers of identical antibodies. And that identical antibodies are also known as the monoclonal antibodies. The name monoclonal antibodies are given to them because they are identical and have single specificity. In this figure, we can see a picture of monoclonal antibodies. What is its history? Who discovered it? Who named it? The production of monoclonal antibodies are invented by the Cesar Milstein and George J.F. Kohler in 1975. Both these scientists were awarded Nobel Prize for their work in 1984. And the named hybridoma was coined by the Leonard Hasenberg. In this slide, we will glance at the three scientists, the two who produced the monoclonal antibody and the one who named it the hybridoma cells. The first one is the Cesar Milstein, and this one is J.F. Kohler. These two scientists produced monoclonal antibodies using hybridoma technique. And this guy is Leonard Hasenberg, who coined the name hybridoma cells for this technique. How hybridoma technique works? What is its principles? In this technique, monoclonal antibodies are generated by fusing B cells with myeloma tumor cells, which grow in cultures. Myeloma tumor cells are generating isolated from the bone cells or tumor cells. The resulting hybrid cell is called the hybridoma. As we can see clearly that B cell fuse with the myeloma cell and the resulting cell is called the hybridoma cell. The hybrid cell has the capacity of antibody production derived from B cell. It can divide continuously by the quality derived from the myeloma cell. It means that B cells are responsible for antibody production while myeloma cells continuously grow. So as a result, the monoclonal antibodies produce cells which continuously growing produce antibodies. By combining the desired qualities of both the cells, which qualities? The first quality is the production of antibody, which are derived from the B cells. And the second quality is continuously growth, which is a quality of myeloma cells. The technology ensures large-scale antibody production of single specificity. In this diagram, we see that it is B cell. This B cell fused with myeloma cell, and the resulting cell is an antibody, and this antibody is named as monoclonal antibody. How monoclonal antibodies are produced? For the production of monoclonal antibodies, we take mixture of fused and unfused cells. Myeloma cells, which lack HGPRT. HGPRT stand for hypoxanthine guanine phosphoribosyl transferase. While TK is an enzyme which is known as thymidine kinase. These both are cultured in HAT medium, HAT medium, and we will discuss it later. What is HAT medium and what role play by the HGPRT or TK. After fusion of the cells, we gain clonal expansion. First of all, we immunized our lab animals, and usually we take mouse as an experimental element. From that immunized mouse, we take antibody forming cells, which is known as the B cells. From the same mouse, we culture tissues, and from that tissue culture, we take tumor cells. And both antibody producing cells and tumor cells fuse together, and they formed a hybrid cell, which is known as the hybridoma. These hybridomas are then monoclonized and isolated for further cultivation. From that cultivation or from that culture, we take antibody producing hybridomas cloned. And that clones are then screened for antibody production. And the whole process is known as monoclonal antibody production. Now we will discuss about the detailed procedure of the hybridoma technique. The steps include immunization of mouse, as we discussed earlier, isolation of spleen cells, production of hybridoma cell, screening of hybridoma cell, culturing of hybridoma cells, screening for desired antibodies, selection and culture of screened antibody. First of all, the mouse is immunized, and the immunization is take place by specific antigen injection against which monoclonal antibodies have to be produced. After 72 hours of immunization, spleen is collected from mouse, and that spleen is responsible for the production of B cells. And from that B cells, we produce antibodies. The B cells are fused with immortalized myeloma cells. The myeloma cells are called immortalized because they grow continuously and didn't die. The fused cells are incubated in HAT medium, HAT medium. First of all, antigen injection is injected into a mouse. From that mouse, plasma cells are derived, and from the same mouse, tumor cells are derived. From plasma cells, we contain B cells, and from tumor cells, we contain myeloma cells. And both these cells are fused together to form the hybridoma cells. And that hybridoma cells are then cultivated, colonized, cloned, and screened to produce monoclonal antibodies. Procedure continued as the hybridoma cells are separated and culture one cell per well. It is called clonal culture because these are monoclonal antibodies. Monoclonal means they are derived from the single source and have specific identification. For screening of desired antibodies, ELISA is performed. Antigens are immobilized in the well and antibodies were transferred so they can bind to antigens. Desired antibodies are grown in mass culture after looking for positive results. Now, dramatically, we can see that immunization. In immunization, an antigen is injected into the mouse, and then Fusion take place, and the fusion of what? B cells and myeloma cells. These B cells and myeloma cells are fused together to form the hybridoma cells. These hybridoma cells are then cultured in the HAT medium. From that HAT medium, desired hybridoma cells are screened by the ELISA. After screening, we have hybridoma screening for antibody of interest. When we have the antibody of interest, then we cloned it. After cloning, the desired antibodies are produced. And these antibodies are called monoclonal antibodies, and that monoclonal antibodies are then expanded and stored for the further use. Now we will discuss about what is HAT medium, HAT medium. HAT medium is an essential part of the hybridoma technique. Without HAT medium, there is no cultivation of B cells and myeloma cells which produce hybridoma cells. The hybridoma cells are selected by using selective media, and that media is known as HAT medium. And what is HAT? It is an abbreviation of hypoxanthine, aminopterin, and thymidine. The unfused B cell will die due to their short lifespan. The myeloma cells synthesize DNA nucleotide by using two ways. And these two pathways are known as the de novo pathway and salvage pathway. What is de novo pathway? De novo pathway refers to the synthesis of complex molecules from the simpler ones. The synthesis of complex molecules from the simpler ones, such as sugars or amino acids, which are joined together to form a complex molecule. As we talk about the salvage pathway, the salvage pathway is pathway in which a biological product is produced for intermediates in the degradative pathway of its own. For example, it is used to recover bases and nucleotides that are formed during degradation of RNA or DNA. In HET medium, the myeloma cells are unable to replicate because the de novo pathway is blocked by the aminopterin in the medium. Aminopterin is the basic chemical which is present in the HAT medium. When the de novo pathway is blocked, then we have only one pathway left, which is called the salvage pathway. So the cell utilizes salvage pathway as an alternative pathway. But it cannot take place due to the lack of HGPRT. As we talked about earlier, HGPRT abbreviated for hypoxanthine guanine phosphoribosyl transferase. For the production of hybridoma cell, HGPRT is very essential. So this HGPRT is contributed by the B cells, which are rich in HGPRT. The salvage pathway is also inhibited due to mutation of thymidine kinase. Thymidine kinase is an enzyme that catalyzes the phosphorylation reaction. The resulting close of hybridoma cells secretes large quantities of monoclonal antibodies. This is B cell. B cell is rich with HGPRT. From these B cells, we derive some cells. Myeloma cells is actually deprived of HGPRT. This contribute 8 cells. These both cells are fused together using PEG. This is the hybrid which contain both the B cell part and the myeloma cell part. This hybrid is then cultured in HAT medium. This is a medium which contain hybrid cells of the hybridoma. After the cultivation, large scale of culture of hybrid cells is produced. These are unfused B cells undergo normal cell death because we know that B cell have short lifespans. So they die normally. These are leftover myeloma cells, and myeloma cells cannot grow in H80 medium because we know that for the growth in H80 medium, HGPRT is very necessary, very essential part. So the myeloma cells lack HGPRT, so cannot grow. If we talk about applications, means where we can use the hybridoma technique. The first application is the serological application. Serological application in which identification of ABO blood group is done. We can also use hybridoma technique for diagnosis. For example, detection of pregnancy by assaying of hormones with monoclonal antibodies, separation of one substance from a mixture of very similar molecules. The other application of hybridoma technique is immunopurification, which means purification of individual interferon using monoclonal antibodies. Further, if we talk about the applications, the further application of hybridoma technique is the therapy, removal of tumor cells from bone marrow, treatment of acute renal failure, treatment of malignant leukemic cells, B cells, lymphocytes, and variety of allograft rejections after transplantation. For this point, we talked about what is hybridoma technique, what does it mean, how it works, what is the principle behind it, what's its procedure. Step by step, we know about the procedures. Now, we will only summarize it in few words. Like hybridoma are cells that have been engineered to produce a desired antibody in large amounts. The advantage of this process is that it can combine the qualities of the two different types of cells. In the whole lecture, we only talked about the two cells, B cells and myeloma cells, which are the actual base of the hybridoma hybrid cells. The ability to grow continually, and this is, as we know, is the quality of myeloma cells, and to produce large amounts of pure antibody, which is the quality of B cells. So these two qualities are fused together to form the hybridoma cells, the hybrid cells, which contain both the qualities, the continuous growth and also the antibody production. Production of monoclonal antibodies, as we discussed, that these are known as monoclonal antibodies because we take it from the same source and have identical and have specificity, which are spread in all the clonal expansions. So this is all about the hybridoma technique. Thank you very much.